Hey everyone, welcome to our final video about materials moving across the cell membrane. This time, we're going to talk about certain types of diffusion that require proteins in the cell membrane. So you will remember that proteins are a huge part of the cell membrane. And the first type we're going to talk about is actually a type of passive transport that is called facilitated diffusion. And that is the movement of specific molecules across the cell membrane through a protein. But look, we're still going from an area of high concentration to an area of low concentration. We're going high to low. We're going with the flow or with the concentration gradient, and it does not require energy. So if it uses a protein but it doesn't require energy, it is facilitated diffusion. Now, you can do facilitated diffusion through a couple different ways. There are protein channels where the proteins just allow materials to go straight through, still going high to low, going with the concentration gradient, passive transport. There are also carrier proteins, and they kind of change shape and move a little bit as they kind of grab materials from outside the cell and bring them in. And those are still going high to low. We're going with the flow or with the concentration gradient. It is still passive transport. But these are both facilitated diffusion because they both involve proteins. Next up, we're going to switch gears. We're going to talk about active transport. Now remember, that's low to high go against the tide. It's active transport. So low concentration moving to an area of high concentration, it is against the concentration gradient and it does require energy, usually in the form of ATP. We're going to talk about a couple different types of this, but check it out. If we were moving from the inside of the cell, low concentration of solute, to the outside of the cell, high concentration of solute, it's going to take energy to move from low to high, to move just like this, low to high against the concentration gradient. It will take not only a protein to help, but also energy in the form of this stuff called ATP, which we're going to talk about coming up here in a couple weeks. Now, there is active molecular transport where molecules move through proteins that kind of act like pumps that require ATP energy, and they go from low concentration to high. If it's low to high, it's against the flow or against the concentration gradient, and it is called active transport. And any type of active transport will require ATP energy. Next, let's talk about a couple ways that cells bring a lot of material in or push a lot of material out of themselves. The first thing we're going to talk about is bringing material into a cell by means of infoldings or pockets on the cell membrane, and that is called endocytosis. Endo sounds like in, the materials are coming in, and as they come in, the cell membrane kind of pinches an area or a vesicle around them and the materials can be brought into the cell. It's kind of interesting. There's several different types, and the types we're going to talk about are phagocytosis, which is cell eating, eating larger molecules, and penocytosis, which is taking in small molecules or liquid molecules. So here's the difference between penocytosis and phagocytosis. So phagocytosis is when the cell takes in large molecules or large particles, like a defense cell in our body could do phagocytosis and take in a large bacteria, actually not a molecule, but a cell, and bring it into the cell. That is a type of endocytosis moving in that is a large particle, and that is called phagocytosis. Now, when the cell wants to take in small particles or liquid, it's called penocytosis, and it takes it in still using the cell membrane to form a vesicle, but it's also known as cell drinking because it's either small particles or liquid, so penocytosis. Next up, we've got exocytosis. Exo kind of sounds like exit. And in exocytosis, the cells release a large amount of material out of the cell. So you start with some material in a vesicle, possibly made by the Golgi body, and then that vesicle approaches the cell membrane, whereas phospholipids join in with the cell membrane, and then it pushes the material out of the cell. That is exocytosis, where the material is exiting. All right. Hopefully you feel like you have learned a good deal about the cell membrane. I'm not going to play this video as part of this ed puzzle, but I would recommend that you watch this video. It is going to be found in the optional or supplemental learning opportunities on Canvas. Thank you so much for listening.